1,000 plus, more than 19,000 manufacturers or importers or license holders have registered in the Central Licensing Authority, that is CDSO. So imagine 19,000 industries have already registered in medical devices also, though the clinical related business in medical device industry are also growing. But when it comes to the medical devices, it is known as clinical investigation. It is not clinical trial, it's a clinical investigation. That is, performance of a medical device needs to be monitored. Again, that is also controlled by the Central Licensing Authority, CDSO, under the provisions of Medical Devices Rule 2017. It's effective from 1st January 2018, but this rule is applicable in our country 2017. So it is one of the historical decision made by our government. We have a separate provisions, rule, regulations for the clinical trial, clinical investigations for medical devices also. Only few developed countries, they have regulation or guideline for medical devices. But again, you as a researcher or as a student, if you are interested to enter into the clinical research domain, you must be familiar with the clinical investigation of a medical devices also. Again, there are IVDs, in vitro diagnostics. There the clinical investigation goes to sensitivity of a medical device. So that also you should be familiar. Again, we have Medical Device Rule 2017 and some guidelines in the ISO 14155. So all these provisions you must be familiar once you are enter into the clinical practice of medicine or medical devices. Medicine, you have a new clinical trial rule. Of course, many of our colleagues, speakers are going to discuss with you. So try to capture all those information, try to interact with all the speakers to get your knowledge and updation. So there are some difference between clinical trial and pharmacovigilance. So that also you should remember, you should know. It may be a funny or it may be a very simple question you may ask. A medicine is tested during the 4 different phases of clinical trial. Why again a pharmacovigilance system is required? So basically, clinical trial is conducted under a controlled or customized environment. You have a very well defined inclusion criteria, patient randomization in clinical trials. You cannot include the vulnerable population in the clinical trials. When I say vulnerable population, there are guidelines. Who are the vulnerable population? Pregnant women, children, geriatrics. So all of them are considered as vulnerable population. So this vulnerable group is not included in the clinical trial. But when a medicine comes to a market, it is being used to all population. There may be different characteristics of a medicine or different adverse events. Those things are not noticed or documented in the clinical trial. So therefore, pharmacovigilance is must to ensure the safety of medicine throughout a life period. So this is the difference between pharmacovigilance and clinical trial. I am sure this also will be addressed. When it comes to pharmacovigilance, India is one of the leading country in pharmacovigilance. We have more than 1 million India specific data. Currently, we don't require to follow any other countries. Our regulatory system is very comfortable position to take any decision based on our own medicines. We don't require to follow any other country's data. Sorry. So that credit goes to the institution like you, because Salem itself, Salem districts alone, around 35 lakhs of population. And 35 lakhs population, to monitor the safety of 35 lakhs population, very few monitoring centers may not be sufficient. Currently, if you look, if you examine the monitoring centers in the Salem districts. We have Mogan Kumaramangalam Medical College or some private institutions. But this is not sufficient to monitor the entire 35 lakhs population. So I take this opportunity to Vinayaga Missions Medical College also can be a part of the system. Already you are reporting, but officially you can also enroll in the system. Not only Vinayaga, there may be so many medical colleges might be presenting here. So I appeal all of them to join hands with the Indian Pharmacopeia Commission to report the adverse events, to monitor the safety of patients. So again, once again, with these words, I congratulate the organizers, Vinayaga Missions Research Foundation, and all the delegates taking part of this conference. Thank you very much and wish you all the best.
Thank you, sir. Your words have truly set the intellectual momentum for this conference. Gratitude is the fairest blossom which springs from the soul. As a token of our appreciation and respect, we are happy to honor our dignitaries with a shawl and mementos. May I now request Vice Chancellor sir to honor our chief guest. Once again, I request our Vice Chancellor sir to honor Thiru N.V. Chandrasekhar sir. May I now request Dr. B. Jayagar sir to honor our Vice Chancellor. I now request Professor Dr. K. Erilvendan sir to honor our registrar. Everyone, this token of appreciation symbolizes our immense gratitude for all your valuable presence and contribution. Knowledge grows when shared. Gratitude grows when expressed. To propose vote of thanks, I invite Professor Dr. K. Erilvendan Sir, Dean, Vinayaka Missions, Kirbandavaryar Medical College and Hospital, Salem.
On behalf of organizing committee, we have to thank our chief guest of today's one, Dr. V. Karaiselvan sir, Secretary cum Scientific Director, Indian Pharmacopeia Commission, and Ministry of Health and Family Affairs, Government of India. Sir, thank you so much for your wonderful keynote address and your presence. Thank you so much, sir. On behalf of organizing committee, we have to thank our guest of honor, Thru N.V. Chandrasekhar sir, the Vice President of Vinayaka Mission Research Foundation. Thank you, sir. This entire conference of about as a clinical trial 2030, the new era, is initiated as a wonderful idea from the Vinayaka Mission's Research Foundation team led by our Honorable Vice Chancellor, Dr. P.K. Sudhir says, his constant motivation and had ideas and inputs to this conference. And just 3 months before he started this initiation, now with more than 850 participants from all over the India. And is also the presentation of the scientific paper. We have restricted around 150 to 175 papers. This is one of the biggest scientific paper posters in clinical trials in India. Thank you, sir, Dr. Professor P.K. Sudhir, sir, Vice Chancellor. And we have to thank our registrar, sir, for his constant support, ideas, and all this conference proceedings for this one, and with Pro Vice Chancellor, sir, Dr. Sabrinathan sir. Thank you so much, Registrar sir. Because of our organizing committee, we have to thank entire Vinayaka Mission Research Foundation, university officials, and we have to say big thanks to our scientific committee led by Professor Arul sir and Professor Jai Chandran and Dr. Sendil Kumar team, who's worked day and night for this wonderful scientific session is going to happen today. Thank you all, thank you guys. Because of organizing committee, we have to thank all the head of the institutions who has given us enormous support and has given a permission for the postgraduate and faculty to participation in this wonderful clinical trial 2030. Thank you all head of the departments. Thank you one more time. Because of our organizing committee, we have to thank all the committees. Like we don't want to take much time because of all the supporting team and particularly IT team and the media press team and all the faculties and participants and postgraduates and supporting staff of this conference. And very, very, very important, we have to thank the professor of principal and professor of engineering college, Dr. Sasi sir, for his contribution as well as his gift for the logistic and infrastructure. Thank you, sir. And if you missed any name, extremely sorry, because the time is you are going to listen this all as 8 keynote spreaders. More than that, we have to thank all the resource persons who has come down from all over the India, particularly, and we have to point out name as from Karaiselvan sir, and Dr. Mujibuddin, Dr. Melvin George, Dr. Kalaivani, Dr. Pratima, Dr. Venkatesh. Thank you so much, sir, for this contribution. And because of organizing committee, we have to thank all the higher officials, controller of examinations, and other people. Thank you so much. Thank you.
Thank you for your sincere words of gratitude, sir.
As we know that this clinical trial, again we want to say give a big round of applause to our Chancellor and the President of our Vinayaka Mission Research Foundation, who has given this enormous idea, and this scientific committee team and organizing chairman, Jayakar sir. Give him a big round of applause, please. Thank you.
Thank you, sir. Having concluded the inaugural session, I now invite the scientific committee to carry forward the academic proceedings. Wishing you all a productive and intellectually enriching day ahead. I request the dignitaries to take their seats off the dais.
Medical devices.
Best wishes, sir. Thank you.
Respected dignitaries, esteemed speakers, faculty members, researchers, and dear participants, good morning once again. With the successful completion of the inaugural session, we now begin the scientific program of this conference. Clinical trials today are no longer confined to conventional models. We are witnessing decentralization, AI-driven methodologies, adaptive trial designs, and increasing ethical and regulatory scrutiny. And the future of the healthcare innovation depends on how responsibly, rigorously, and intelligently we conduct clinical research. And this platform brings together regulators, academicians, clinicians, and industry leaders. We are not here merely to listen. We are here to reflect, question, and strengthen our approach to research. With that note, we commence the scientific session for today. Technology is no longer merely supportive. It is redefining how clinical trials are designed, monitored, and executed. I take great pleasure in welcoming our distinguished speaker, Mr. C.S. Mujibuddin, founder and CEO, Clinosol Research Private Limited, Hyderabad, who will be enlightening us with his expertise. Sir, we welcome you. I also extend a warm welcome to our respected chairperson, Professor Dr. Rangabashyam, Medical Superintendent, Vinayaka Mission Kripananda Varyar Medical College, Salem. May I request the chairperson to kindly introduce the speaker and conduct the session.
For the opportunity to sit as a chairperson for this session, and thanks to the organizing team. So the clinical trials are the need of the hour in our country. So it is going to serve this mankind by giving new drugs for the usage and also to assess the efficacy of the new drugs. So it's better for all the medical professionals and the pharmaceutical people to get involved in the clinical trials to help and serve our mankind. And the topic of this session is about advancing clinical trials with artificial intelligence and decentralization. The artificial intelligence is going to rule the next 2 decades in our country. So it is getting infiltrated in all the fields. So the clinical trials are not the exceptions for the artificial intelligence. And this knowledge of artificial intelligence is going to make the clinical trials easy, and it reduces the time consumed for doing the clinical trials. So the busy practitioners also may get involved in the clinical trials without wasting much time by using this artificial intelligence. To enlighten about this topic, we have our esteemed speaker, Professor C.S. Mujibuddin, is the founder and CEO of Clinosol Research Private Limited, where he leads pioneering initiatives in the clinical research and healthcare innovation. He also serves as Associate Director of Sai Ganga Panacea, Digital Health Sciences and Strategic Partner, contributing actively to the advancement of digital health and technology-enabled care. Bridging academia and professional practice, he is a professor of practice at St. Anne's College of Women, Medipatnam, where he guides and mentors aspiring healthcare and research professionals. In addition, he serves as a director at Rapid Medics Hospital, playing an important role in strengthening patient care, clinical systems, and quality-driven healthcare delivery. With a distinguished career spanning research, education, and healthcare leadership, Professor Mujibuddin continues to foster innovation, collaboration, and excellence in the medical and digital health domains. So over to the speaker.
Okay, good morning. Good morning. I am not able to hear you. Let's try once again. Good morning. Last once again. Good morning. So my ears are not working or your voice is not working. Let's try once again. Good morning. Thank you so much. I would request all the students to settle down. That does not disturb my session. I'll give exactly 30 seconds of time so that everyone settle down. 30 seconds. Thank you everyone. Please get settled down so that I can complete my session quickly. Okay, wonderful. So I have my slides. So you have 2 good news. Can I tell good news before I start the session? Yes or no? Yes or no? So today you are having a very crisp session. It will not go more than 30 minutes because I have a flight. So I have to go and catch the flight. So 30 minutes I will complete your session. That's point number 1. Point number 2, I am today fasting, so I'll not ask any questions to you so that you don't ask me questions. I can run away from here. So these are 2 good news. I'll be very crisp, short, and this topic is very, very, very challenging. AI and decentralized clinical trials. I'll not take much time. So this is me. As of now, if you see a brief snapshot about me, what I generally do, what is my profile. So it's about 3 profiles I have as of now. I am a founder and CEO for Clinosol Research, and I am the associate director for a medical device company, and I'm also a director for Rapid Medic Hospital, where we develop a lot of devices and also apps for healthcare sector. I'm a professor of practice, and this is my achievements. We have already reached to a students of 50,000. On average, I give nearly 150 to 200 sessions every year. So that's more important. And then I have more than 19 years of experience, and I have got back-to-back 13 awards. This is my profile. And yeah, so this is more interesting slide. First of all, the previous speakers and chief guest has spoken very nicely about clinical trials, because most of molecules needs evidence. Without an evidence, we can't bring the molecule into the market. We have to test it in very controlled and customized environment, and it has to go into market. But the earlier scenario was different, and now scenario was different. Earlier it was taking around 15 years to 20 years, but now it is only taking 4 to 5 years. How can that time has been reduced? What are the different strategies we have used to reduce the timeline? Still we are understanding the importance of the patient safety and efficacy. So that's the first one. What is this traditional trials? Why these are struggling? So I think everyone knows there is a theoretical aspect, that what are clinical trials, why clinical trials, how clinical trials, and all those answers will come. So the traditional clinical trials have some barriers. So because a patient has to come to hospital, and the hospital should have this sites, and they have to conduct clinical trials. So the first one is geographical barriers. Only one place, one location, if you conduct a clinical trial, you might not show efficacy. You have to test it at multi-center sites, different location sites. Earlier it was having so much challenges because there was no electronic transmission, or there was no more electronic data capture. The second one, the manual errors and data source. The huge aspect, I would like to inform all the B.Pharm or M.Pharm or PharmD students, your future is only one word. That word is called as data. Everything will be spoken on data. So data-driven decisions, or data-driven clinical trials, or data-driven adaptive trials, or whatever it is, if you have enough data, you can perform more clinical trials. Whatever you give, for example, if you give garbage, it will come out of garbage. If you give a clean data, it will process a clean data and come up with more understanding that how really drug is working and which categorization of patients are working. So that's data soil and manual error. That's the second thing. And the third one, the hospital selection. Already we spoke about it. How many of you before coming to this really knows about clinical trials? The awareness of clinical trials was less. The regulations were, knowledge about regulations were very less, and about site selections, hospital selection was very, very less. That's the third one. And the fourth one, regulatory complexities. Sometimes it happens that regulators are giving more strict regulations, stringent regulations to safeguard the human beings. Either it is USFDA or CDSU or other regulations. These were all issues with traditional clinical trials. Now here comes the first chapter, which is called as AI and decentralized. There are 2 words. First one is AI, and the second one is decentralized. And remember, AI, what we are using for engineering is entirely separate from AI, which we are using for clinical trials. Don't club both. So it's entirely different process, because we are working with human beings. Okay, now this is a very interesting story. What exactly is decentralized clinical trials? Now I'll ask you a question. Patient will come to hospital, or doctor will go to a patient? What method we are using? So not energy and efforts, and also it includes your finances. Why can't we make every patient home as a hospital? That's where the picture got changed. So now we are using multiple technologies, and also we are using multiple systems where every patient home can become a hospital, and he does not need to come to a hospital to participate in clinical trials. So now future is, you don't find a patient searching for you and coming. You have to go and search a patient and enroll the subject into the clinical trials. That's where the picture comes change. That's decentralized clinical trial. First one, e-consent. Earlier, the consent form was a big paper, 10 pages, and patient has to read everything and understand. Now it entirely got changed. It's all called as electronic and video documents or videos or gamification, or a patient can click on any video to know about the drug or disease or clinical trial or investigator or all those things. So now it's completely zero paper, no travel is needed. Patient can participate in clinical trial by just attending in a video consent. That's the first point. The second point, I think very interestingly, our chief guest also spoke very nicely about the AI summit which is happening. One of the most interesting thing which is happening in AI summit in Delhi is wearables. The future is all about wearables. Understand it very clearly. Future is not about diseases or drugs. It's about wearables. How you can take the wearables into consideration. Wearable is not just what you are thinking. Wearable can be a watch, wearable can be a ring, wearable can be a glass. Very recently, AI Summit also, there is a one glass which has been developed. Whatever languages you are talking also, if you wear that glasses, it converts that into your local language. That's where the innovation is coming. So you just have to understand that the innovation is coming in such a way that the clinical trials are growing so much. So the second one, very important one, is wearable. Patient don't need to come to doctor to give a vital signs. There is no requirement of a doctor to get a BP or a heart rate or a blood pressure. How patient can wear a wearable and give the data, continuous monitoring is being done. I think most of you guys know that there is a continuous glucose monitor. Everyone is using a patches. They put a patch and then continuous glucose is monitoring. That's the second one. The third one, very interesting one. Now the pharmacist directly how they can talk to a patient. Say for example, patient is remote tribal village. How can you dispense a drug to a pharmacist? Drone based dispensing. How can you use a telepharmacy? Virtual mode of convincing a patient that this is how you have to take a drug, this is the rational use, this is what you have to avoid, this is what you have to do. All those things are called as tele. Word called as tele. NMC has already given regulations. Also Pharmacy Council also has given regulations for conducting telepharmacy and telemedicine. Both are acceptable now. So doctor can be convinced the patient in regards to your tele. All these three things make patient empowered, that patient does not need to come to a hospital to participate. That's what is the future which is called as decentralized. Patient will not come to a hospital, but every patient home will become a hospital for you, or patient will become more empowered and he will be engaged. He does not want to exit from the trial. Because most of times patient will exit. If he does not find interesting, he will say I am exiting. If you make sure that he is engaging with the data and you say that your all details are being captured continuously, then he makes sure that he continues into trial. Yeah, so this is one of the interesting story. Now I said about decentralized. Now how AI is making decentralization possible. How artificial intelligence is making sure. Okay, how AI is making decentralization. I think very recently, I think 2 days back I read one article. And everyone is speaking now about AI uses in clinical trial. The first and foremost aspect, site and patient intelligence. Remember, most of drugs will fail because you don't find the right patient, you don't find the right hospital to conduct a clinical trial. You might have a wonderful drug, super good medicine or device or vaccine or cosmetic or whatever it is. But if you don't have a right patient to test it, you can't prove that it is having efficacy and safety. That's where the first point is your identification of site and also identification of right patients. That's what you call it as patient identification. I'll ask you a question. How do you identify a patient? Now currently, doctor is responsible to identify the patient. Patient comes, they discuss and they identify. But now there is one more system which is coming, which is called as EHR and EMR. Electronic health records and electronic medical records. Now, one organization is there where it is collecting all the medical records of the patient. Now, I am going to that organization and saying that, boss, listen, I want this inclusion and exclusion criteria. Can you pick up the data and give? They will give you the data. They will give you a whole patient who fits into your clinical trials. You don't necessarily need now manual process to identify a patient. That's what is the future which is called as EHR and EMR identification of patient, because most of the trials fails not to identify the right patients. The second one, remote trial conduct. So this is most important. The machine learning powered models, which can make sure that which patient is having which serious adverse event. Because we are putting a lot of variables, we are putting a lot of health apps in the phones. So now future is not about RX, it's about DDX, digital therapeutics. Doctors does not give only medicines. Doctors gives one medicine, one software, one app onto you. So that's where understanding of the subject patient data to give further treatment or plan of treatment or changing of the treatment is more important. So that's the second one, remote trial conduct, including either it is telehealth or it is ePro or it is virtual visit. Patient does not need to travel to a hospital unless and until it is very serious conditions or it is very careful assessment which has to be done by the doctor. So if it is not there, then doctor can give his counseling or consulting over the phone and he can continue doing that. So that's your virtual visit. And also there is a ePro, which is called as patient report outcome. Remember, in future, you will not ask patient is getting treatment or not. You will ask how comfortable, how happiness index, what is an outcome of the patient. If he is happy, your drug is good or not good is also okay. So he should continue the medicine. The patient should continue the medicine, or else there is no point in taking a medicine and giving a medicine for the treatment. There is no efficacy saying that disease is going or disease is coming. It's about outcome of the patient. So that's the second one. The third one, very important one, which already have said that data quality on the scale. The highest complex problems are coming with data quality. Because the sources of data has increased. Now I said so many stories. EHR I said, EMR I said, telehealth I said, ePro I said, wearable I said, that I said, this I said, electronic data I said. How many data are getting now? This much data is coming not only from patient, but from the labs, from ECG, from data machines and from wearables. So much of data is coming. How can you all this data bring into understandable data and clean and validate the data? Making sure that it is readily usable data, because the data is so complex. So how can you do that? Data quality on the scale with NLP and also automated pipeline is more important for you. Okay, so now first point, how AI can be used in site selection and hospital selection. I don't know whether Vinayaka machine is doing or not. I am not discussing about that, but I am sitting at Hyderabad and understanding that which hospitals are doing. Now my consideration is in Vinayaka machine also and XYZ also and YZ also. How I can use AI to pick the hospital and site to conduct a clinical trial. That's the first point. So this is the challenge. The first challenge is site selection is always wrong and it has a costly mistake. Identification of right investigator and right patient is always a challenging. But the solution, very clear solution, machine learning, make sure that historic data analysis of EHR and EMR data and the previous record of the investigator, that how many clinical trials are being performed and how many clinical trial data is being submitted. Remotely, you can pick up the site to conduct your clinical trials. That's a chapter 1, which is smarter site selections. And this is one of the interesting story. Site feasibility in practice. Whether we have picked the site, now we are having question, is this feasible or not? They have resources, they have facilities, they have time, they have X, they have Y, they have Z, and they have previously no black spots. This is a feasibility now. The biggest question is the feasibility. How can we take this feasibility into consideration? So this is one. The first one, EHR and registry mining. See, understand, there is always one story. Say for example, how do I know about you? If I know your Facebook and Instagram, I can know about your character now. I can go and do data mining of your data, that how you are putting the data, and then I understand what exactly is your character. The story here is called as data mining, going into deep and deep of the data, understanding what might be my solution based on this information. That's the first one. Second one, investigator profiling. The investigator who is coming into clinical trial has a right qualification, training and experience, and also has a time to conduct a clinical trial. That profiling we can do it, the doctor. The third one, feasibility scoring. AI can put up the scores and say that if you take this site, the chances of pass is this. The chances of continuing this trial is this. The chances of fail is this. So that's your flexibility or feasibility scoring. And last one is dynamic re-ranking also it can change. At any point in time, it will take huge amount of data and answer about the data. AI is all about data. understanding about data and prediction of data is more about AI. So this is the first one. This is a very interesting story. Now everyone wants, including doctors, pharmacists, nurses, everyone wants one person. That person is called as patient. Okay, so now how can you recruit a patient in clinical trials? That's the most interesting, because if your drug is super good also, if a patient is not there, then you can't test it. So how AI plays, already we have discussed about this. First one is EHR based modeling or machines. This is the first one. NLP exact matching can be done. NLP exact matching can be done for that particular patients, picking up the EHR and EMR, because lot of data is there for the patients. The second one is digital and social reach. This is very interesting story. AI makes sure that digital advertisements right to the patient, but this has a complex, because regulation clearly says that you cannot run an advertisements. You cannot run an advertisement in clinical trials. But how can you make sure that digital availability for the patients, how you can take it up is more important. That's your social and digital. Third one is protocol optimization. Understand, earlier it was bench to patient, now it is patient to bench. Earlier you used to go into a lab, discover a drug, come to a patient. Now you have a patient, identify a disease, then come back to a lab and identify the drug which matches this. So I think previous speakers or previous chief guest also spoke about precision medicine. One dress, one blazer does not suit for everyone. How can one drug does suit for everyone? How can you identify that all kind of social, physical, genomic, omic data taken into consideration along with combination of medicines which are taken into consideration along with your demography data, how drug can exactly act on it? That's called as optimization. How can you make sure that your protocol is as per the fitting of the patients? Is something AI can do it. So that's the first chapter, the patients. This is the advantage, evidence from the field. These are the advantage of AI in patient recruitment. First is the faster screening, AI assisted, nearly 72%. And enrollment speed is more than 50%. And diverse patients, because you can't take one people. So now I think today morning also so many people spoke about 800 people have registered and 70 institutions. It is called as diversification. We're not discussing about one institution, it is speaking about multiple institutions and multiple genders, multiple races, multiple qualifications, multiple things. That's called as diverse patient population. So this is the advantage. This is the second chapter, very interesting. Patient once come into trial have to be engaged and stay in trial till the end, or else there is no point of continuing a trial. So most of patients will fail, and most of patients will say that I have a doubt about this clinical trial, I don't want to continue. How can you engage a patient to continue into clinical trial at the end? So that's AI in patient recruitment or patient retention. So this is keeping the patients engaged and on study. The first point, you can do a dropout prediction. I don't know how many of you know. So there is so much of AI now that I was speaking to one of the technology people and they were saying that on a call center, based on your voice and based on your words, we know that how much you are showing interest to take that product or service. So that how much we are bringing is, if you are calling to a call center saying that I want to know about this product, based on your voice, terms, words, and the gap you are taking, we understand that whether you are really interested or you are dropping out. That's called as AI prediction. The same way we can implement in your dropout prediction. So based on how many times the patient is really engaging with the investigator, either through WhatsApps or through mobile or through how good he is communicating, based on it we can predict the dropout. And before he goes into further last, we can take that patient and counsel the patient and answer all the questions of the patient. That's called as first one, dropout predictions. Second one, most important thing is we forget. Because of all technologies, we forget. That's called as personalized reminders. Now, more interestingly, there is one word which is called as agentic AI. AI and agentic AI is more advanced. I'll just tell one small story here. Say for example, patient has to come to a visit on 2nd of February. He forget. We gave a reminders, he forget. So automatically, now AI what it does, it gives a new dates. But agentic AI does not does that. So what it does, what was a mistake? We gave a reminders, patient forget. So now second time what agentic AI will do is, they'll book a Uber cab on 8th. And Uber cab will go and call the patient, still he picks the call. And he picks the call, Uber picks the subject and go to a hospital, nearest hospital, and then site is been taking the assessment. Then Uber is waiting for you and he will take that patient to the home. Now it automatically updates the next visit dates also. You don't need a human now. It automatically updates that next visit, how it have to happen, and next visit reminders it will give. Now you don't need to go and have an Excel saying that when patient is came, how much days difference, when patient has to come. All this are not required now. It's more about reminders. Everyone has one of the most important activity, personalized reminders. The third one, home-based visits. Not necessary that patient has to go to a 40 kilometers away to a hospital. If there is a no serious cases, he can go to a nearest clinic and take that data and submit it into the electronic systems. And home visits also can happen with the respect to your engagement. So these are all engagements, and we are bringing chatbots also. Now chatbots are like our best friends now. And if you give garbage, it will give garbage. Understand? If you ask chatbot that some worst question, next time it will give worst answers only. So because I think very recently, I don't know how many of you know this funny story. One time ChatGPT, one lady was asking some question and ChatGPT gave wrong answers. This lady got so frustrated and she scolded ChatGPT, saying that you idiot, you don't know this, that, all those things. Then next 2 days, she asked that what are the places to watch in this particular location because I am visiting. ChatGPT gave the answers, and then these people went to that all locations, they came back. But finally we understood that ChatGPT gave the worst last 10 options because ChatGPT was being scolded. So later on we understood that this is what happened. And I think 2-3 days back, one of the person was writing an article in ChatGPT. ChatGPT understood that he is doing a wrong research. ChatGPT itself wrote an article and submitted to one publisher saying that this fellow is doing all worst publications. So now I don't understand how ChatGPT is taking our information and how it is giving to others also. So because if I give some slides and say that can you modify this slide, it is taking that slide. But whom it is giving the slides, I don't know. So it might give to someone else also. So much of complex information and things are there. Yeah, so this is ePro platforms, the patient report outcome. Remember, patient should be empowered. Future is patient should be empowered. Patient should not feel that I am separate from the clinical trial. So there is a word which is called as patient-centered clinical trial. Patient should take into the consideration, patient voice should be heard, patient assessment should be taken into consideration. That's the first one, ePro. Second one, I already have spoken. Future is all about wearables and sensors. There is no other words. Already I think our chief guest has spoken about devices. It's all about wearables and sensors. And the third one, telehealth visits. Not every visit patient has to physically come. Some visits he can come on phone or on Zoom call or XYZ. And I already have said AI chatbots. Chatbots are the future. So chatbots are becoming more important in our life. Yeah, this is chapter 5, which is, I already have spoken, data. Data. Every time you forget about everything, but you remember only one thing, data. What is the data you are producing? What is the data you are analyzing? What is the data which you are predicting? So the most important word is the data collections and data analysis and data validation and data predictions and archiving of data also. So that's the future of your data cleaning. Yeah. So the AI driven data quality management, because most of us ignores about data. The data which should be clean, because regulators are already here. Now they only see the data, but they don't see the patients. They don't see the procedures. They don't see anything. They only see one word. What is that? What you have submitted the data, they will review that data and they will approve the clinical trials. But they don't go and see that who is the patient. You bring the patient and I will ask some questions to the patients. The data has been given so advantage, and AI driven data quality management has been done more and more. And remember, there is one word which is called as risk-based monitoring. You have 10 risks in clinical trial. Which one to prioritize? That's called as the risk-based monitoring. So the biggest risk has to be given more pressure. The biggest problem has to be given more time and efforts so that the rest of things can be taken into later categories. So that's your risk-based monitoring. So this is the risk-based monitoring, AI in action. So many issues are happening because of human resources constraint or because of regulatory knowledge and so many things. Now, centralized statistical systems. You don't need a monitor to go to a hospital and review the data. He can sit at one central location, all the data is uploaded and he can review the data. That's called as central data monitoring and statistical consideration. And there is one more thing, automated edit checks. Say for example, patient signed ICF today, but patient got adverse event yesterday. This is invalid data. But you don't require manually to check this all invalid data. Forget about this works. You will not have this kind of works now. Automatic edit checks will fire saying that, boss, listen, your adverse event has happened before ICF, it is not possible, can you correct it? And system will ask. And site will understand and answer and system will close. No human being work. Understand, human being work will become more critical with the use of AI. So that's what it has to be understood. And query prioritization. Already I have said, there are 100 queries. Which query is more challenging? Now say for example, there is a patient who got a serious adverse event, but there is no treatment. I got doubt now. You got a serious adverse event, how can you don't give a treatment? As per GCP, you have to give. That query has to be prioritized when it is compared with ICF or AE date. Might be they have made a small mistake, but SAE they can't make a mistake. Every patient who got an SAE has to be immediately treated. So those kind of prioritization can be done. And the fourth one, regulatory compliance. Remember, no paper studies going forward. No paper studies going forward. It's all about electronic system, electronic signature with a username and password. How you can make your data secure. So now say for example, you understand or not, privacy concerns are more important in India now. And you are working with the patient data. And your patient data has to be secured very much highly or else you'll be in a big problem. Okay, so this is one session which I always feel that this session should be understood. Which jobs are safe for you? Understand, it's your data analytics and data visualization jobs. I would ask you guys to learn about how data can be visualized in a pictorial representation, because one picture speaks 1,000 words. It's not 1,000 words which speaks about one picture. So it's all future is on data analysis and visualization. So this is raw data with visualization. This is the different softwares are there. I don't want to go into it, but here so many systems are being given here into it. So advanced AI techniques, the first one, predictive analytics, the second one, biomarkers discovery, third one, safety signals and subgroup analysis. Yeah, visualizing trial data. Visualizing trial data for decision makers. Understand, once you have enough data, historic data, visualize that data and predict it whether I am going in a right way or not. Remember this, there is no fixed clinical trials that I start now, after 6 months I will stop my trial. No. Every month you stop a trial, you collect the data and you analyze and visualize that. Can I go in a right way or should I modify my protocol? Should I change my optimization of subjects or should I change my medicine? Or should I stop it? Because after 6 months you are coming and telling that drug has failed. And 6 months patients are thinking that drug is working really. So how can you make this kind of environment is more that predictions and adaptive trials. Forget about fixed trials. See, now your history says 1:1 drug. Paracetamol is good or diclofenac is good. Now the future is not like that. Future is like paracetamol is there, but paracetamol is only working on fever or it is working on cancer or diabetes or something else also. One drug, multiple diseases. Multiple drugs, one diseases. Multiple drugs, multiple diseases. So complicated designs are coming into picture that how you can make it very quick. Adaption of this clinical trial is important. So this is visualizing of trial data for particularly your aspects. Additional AI application in trials, these are protocol design optimization. Now this is wonderful story. Now I don't know how many of you use ChatGPT in a proper way. If you go to a ChatGPT and say that can you design a clinical trial for a diabetes, it will give 1 page of protocol. and once that take that protocol and add some of your manual information and then upload it back to it, say that NDCT rules, NDCT 2019 rules, you download and upload that also to a ChatGPT, say that as per NDCT can you develop your protocol. Now ChatGPT will go and read all NDCT and also read your manual protocol and give one more designed protocol. Now I have more questions. That is this design protocol is as per GCP, I will download ICH E6R3 also. And I will upload that document also ChatGPT saying that, can you now check that, is this my protocol is as per your GCP or not? Then it will give all concerns that these are the places which you are deviating as per GCP. Now you go and ask, as per this protocol, how much time I will take to complete this trial. It will give how protocol can be optimized also. So the future is all about how the protocol design can be optimized and how you can analyze the existing data and do it. So that's a part one. The second one, e-consent and regulatory automations. This is more important. See, we can speak so many things in these conferences, but regulators also have to accept the process which you are doing it. It's not that I speak and go and then regulators are saying that no, this cannot be accepted. So the e-consent has to be accepted, and also there are some regulators which has to be optimization. So because regulatory aspects have to be taken into considerations and submissions have to be done by immediate aspects. So this is real world data. Remember, the future is not about the clinical trial data, but it about real world data. Now doctor is having huge bunch of data that he is taking from the patient. How can he analyze this data and give more data to the sponsors saying that, boss, listen, there are some unacceptable situations where we missed it. Can you take it up? So that's called as your real world evidence and AI. So these are the key publications. These are more evidence publications. These are AI based clinical advancements, and these are the research themes. And these are the challenges. I'll not take much time. Within 1 minute, I'll close this. The challenges, the first one is data privacy and security. The biggest question is how ChatGPT is taking my data, I don't know. So how AI is taking that data and how it can use it, I don't know. The privacy and security concern. Second one, digital divide. So not necessary that everyone will use the way I am telling the stories. It might be some people who are uneducated, illiterate. How can they get the access of internet and all? Digital divide is more. And algorithm bias. Remember the ChatGPT, how it works for me, it will not work in the same way to you. So that's called as algorithm bias. And there is always a black box. And I think our chief guests have spoken about sensitivity of devices. So we really don't know ChatGPT is giving right answer or AI is giving right answer or not. There is something which is called as black box. We really don't know how it gave that answer also. If we ask how you gave this, I don't know, but I gave this answer. If you are interested, use it, or else don't use it. And the third one, regulatory validation. Regulatory validation has to be done very carefully. So this is about DCT and AI in clinical trials, particularly. In NDCT, CDSU has put risk-based monitoring, and Ayushman Bharat has given digital machines, and also the India unique opportunity has been there. So remember what coming up next, AI protocols are coming up next, adaptive platforms, regulatory agents, and also continuous learning is there. So these are the key takeaways. AI accelerates at every stage, including taking the patients, selecting the sites, conducting a trial, submitting the data, getting an approval, and going into market. Every stage AI can be implemented. Remember, don't learn AI without understanding the clinical trials. First go into deep of your pharmacy, then learn AI. Don't go and learn this AI I have learned, but I don't know how to utilize in pharmacy. The point here is you need to first go into deeper of your area, then learn AI and implement that into our pharmacy. The second one, evidence is here. And the third one, decentralization, democratize the clinical trials and act now. With that, I wanted to personally thank our Jaikar sir who has called me for the session. And thank you so much for patient listening. I am open for questions, but due to time lag, you can contact me. My LinkedIn is Mujibuddin Sheikh. In LinkedIn, you can connect with me. I'll definitely answer all your questions. So with that, thank you so much. Presented at Vinayaka Mission Research Foundation, Tamil Nadu. And I'm dedicating this session to Jaikar sir, because Jaikar sir is always my mentor and guru. Thank you. With a round of applause, guys. A big round of applause.
So thank you very much, sir, for your informative speech. For the want of time, you can post your questions in the mail. Thank you so much.
Thank you, sir, for such an insightful and practically grounded presentation. May I request Professor Dr. Rangabhashyam to present the memento to our speaker. Thank you, sir. May I also request Dr. Rajaram, Chairman, Clinical Trial Monitoring Committee, to present the memento to our respected chairperson. Thank you, sir. Participants, kindly note, participants are requested to submit their scientific presentations in their respective halls before the scheduled session. We now move on to the next session, keynote address by Dr. V. Kalaiselvan. Dr. V. Kalaiselvan serves as the Secretary cum Scientific Director of the Indian Pharmacopeia Commission under the Ministry of Health and Family Welfare, Government of India. He holds a PhD in Pharmaceutical Sciences from the University of Delhi and has played a pivotal role in strengthening pharmacopeia standards, drug safety systems, and regulatory science in India. He was instrumental in conceptualizing and implementing the Pharmacovigilance Program of India and also the Materiovigilance Program of India that significantly strengthens national surveillance systems for medicines and medical devices. His contributions have also been recognized internationally, including acknowledgement by the World Health Organization for advancing global pharmacovigilance initiatives. With over 130 research publications and extensive leadership in regulatory science, he brings both scientific depth and also policy vision to today's theme. So, we are honored to have you with us. The stage is yours for the keynote address on future and innovations on clinical trials.
Thank you, and again it's my pleasure to stand before you. So, in continuation to my inaugural address, there are a couple of other examples during my experience which I would like to share with you. A lot of things I have covered under the clinical trial, artificial intelligence, etc. But there are some practical, examples or practical experiences I would like to share with you. Now you might be understood, overall about clinical trials and post-marketing surveillance, right? So as very well said, clinical trials are conducted under controlled and more systematic manner. You have very well defined inclusion criteria, exclusion criteria, all those things. But when it comes to the post-marketing surveillance, after the product is launched, it is being used or applied to all population. There might be a chances of more number of adverse drug reactions or more number of characteristics of a medicine may be exhibited. So there are very well defined examples. I would like to share only 3 examples what we are facing currently and what we are handling. Number 1, sodium valproate. I am sure you all might be aware of sodium valproate. It is as an anti-epileptic medicines. But during the clinical trial, it was not noticed or it was not documented that this medicine is going to cause birth defect, like thalidomide, right? When the thalidomide was introduced, birth defect was not documented. Similarly, sodium valproate, at the time of clinical trial, there was no evidence of birth defect. But in the current practice, after the launch, huge number of reports, even India specific, our own population also, reported this birth defect as one of the issue associated with the sodium valproate. Now this is the importance of pharmacovigilance. So clinical trial, of course, it is required for a short period of time as per the regulation, but when it comes to the market, there may be more number of adverse drug reactions. So now this is the problem with the sodium valproate, and we as a pharmacovigilance, IPC as a pharmacovigilance custodian, what we did, we have asked all the neurologists as well as other stakeholders, it should not be given to the childbearing age group women. It's a kind of advisory, or it's a kind of message through the regulator or through the pharmacovigilance system. It is being circulated all the community that sodium valproate not to be given childbearing age group of women or pregnant women. The doctors should go for alternative drugs. So this is one of the example. Now you can realize, you can convince me, if anything, to be monitored during the post-marketing surveillance. Another example, second example which I would like to highlight here, pregabalin. Pregabalin during the clinical trial, there was no report or no evidence is available regarding the abuse or dependency of a pregabalin. Now, huge number of reports, India-specific reports, of course globally several reports are available that pregabalin is causing dependence. But this was not captured during the clinical trial. Therefore, continuous monitoring is required. Now, what we did as a system or as a responsible organization, in fact, the government of India, recently they are going to come up with some gazette notification for pregabalin. They have asked our comments, particularly whether pregabalin can be shifted to or can be put under the Schedule H1. So, the Indian Pharmacopeia Commission strongly submitted a proposal, not only H1, it should be put under the Schedule X. So this kind of our own evidence, own data, we have submitted to the government. It needs to be changed according to the data available. So these all were not captured during the clinical trial. And third example, very well known drug, paracetamol. Of course, each and every one of us might be taking daily basis, or our family members are taking daily basis. Now emerging evidence that there are autism, paracetamol associated with autism. It is not only it is not our India specific reports. Globally it is coming out. Even you might have listened that US President's statement couple of months ago, about paracetamol autism. He himself declared that autism is associated with paracetamol is emerging in US. So these all are the issues, concerns. We have to monitor the safety throughout its life period. So clinical trial is only for a limited period of time. We as a responsible person, responsible stakeholders, we have to monitor the safety throughout its life period. So now these three reports, if you take these three examples, there are many more examples like this, but I thought of telling you these three examples. We have huge data, and some of the data are reported from the Salem region also. That is what in my beginning, in my opening address also I mentioned. This is not sufficient. Few monitoring centers will not be sufficient to monitor the entire 35 lakhs population of Salem districts. We require to have more number of monitoring centers to come up, more institutions to join hands with IPC for monitoring the safety of all these issues. So this is one of the message which I would like to share in continue with my previous talk. And then of course career perspective, wherever I go, I used to address to the students, the researchers, what about the career opportunity in pharmacovigilance or clinical trials? As I mentioned, clinical trials or pharmacovigilance are considered as one of the regulatory requirements. Pharmacovigilance also a part of a regulatory system. As per the notification, Gazette notification 2017 March, every industry will have a pharmacovigilance unit, and it is being monitored by the trained and qualified pharmacist. So you can imagine the career perspectives for the pharmacist, those who are going to work in the clinical trial or pharmacovigilance system. So the clinical trial knowledge alone will not be sufficient. You require to acquire adequate knowledge about the post-marketing surveillance and pharmacovigilance. And again, I am very happy to share with you that Indian Pharmacopeia Commission is the first government organization to create career and job opportunities for the pharmacist to engage in pharmacovigilance. Accordingly, the recruitment rule or amendment have been placed. So there are a lot of career opportunities for the pharmacist or PharmD professionals, those who are interested to join with pharmacovigilance and also clinical trials. And now another area, medical device. As I mentioned, more than 19,000 industries have registered in India alone. Here also there is missing, some ecosystem was missing. So Indian Pharmacopeia Commission was taken a proactive step. When I say ecosystem, we have a regulation for medical device. We have a policy for medical device. Now 19,000 medical device industries have registered. But where is the skilled or human resource, trained human resource or workforce for medical device? So there the ecosystem was missing. We require qualified, trained manpower also in order to fulfill the regulatory compliance. So what we thought, we approached Pharmacy Council of India in 2019. There is a need for inclusion of medical devices or material vigilance in the educational curriculum. Then only the complete ecosystem will be addressed in order to meet the regulatory requirements or to ensure the patient safety. Now, the Pharmacy Council has taken up an initiative. They are going to introduce the medical devices course very soon. So again, this ecosystem is going to be addressed fully. There also, you have a lot of scope and opportunities for monitoring the safety of medical devices. There are a lot of job opportunities are going to be created. So this is the additional message for all the researchers, students, those who are sitting here, for your career perspective. This is one part of safety related activities I have covered. And second one, I have couple of slides related to quality, because it's the right time to sensitize or to update you about the developments at Indian Pharmacopeia Commission about the standards. The quality and safety both should go together, then only the patient care or patient need needs to be addressed. So I have couple of slides for you regarding the Pharmacopeia, Indian Pharmacopeia. Why the Pharmacopeia is so important for all of you? Because even the clinical trial or pharmacovigilance practices, you might observe some of the lack of efficacy or product quality related issues where there is no product effect or therapeutic efficacy is not delivered because of quality. So quality is equally important in order to ensure the safety. So recently the Indian Pharmacopeia, again it is one of the mandate of Indian Pharmacopeia Commission. It is being introduced, launched recently. But here the message I would like to convey to the young youth, that so far around 95% of the monographs or standards are industry driven. The standards or specifications are contributed by the industry stakeholders. I take this opportunity to say the students and researchers should actively participate in the monograph development process. We have defined inclusion criteria for developing the monographs. So you must contribute in the existing monograph revision or upgradation as well. So there are so many feedback or inputs daily basis we are receiving, that all from the industry. Rarely one or two technical comments or inputs received from the academics. But you have a huge potential and responsibility in implementation of these pharmacopeia standards. So I take this opportunity to use IP monograph, and maybe 5 years, 10 years down the line when we come up with next edition of Indian Pharmacopeia, I would say the contribution or support from the academic institutions or researchers also should increase rapidly. So you must understand, while developing the pharmacopeia standards, it is not only India specific requirements. We have also taken care of the global requirement. As the Indian Pharmacopeia Commission is being recognized globally, our responsibility is not only to look at the India specific need, we are also focusing on the global demand where the pharmacopeia standards needs to be adapted or upgraded. There are 21 new monographs which are antimicrobial and anti-diabetic and anti-cancer, which are included in the Indian Pharmacopeia current edition, that is 2026. If you look at the current edition of Indian Pharmacopeia, one of the significant initiative or steps taken by the Indian Pharmacopeia Commission is we have introduced the standards for blood components, which is not available in any other pharmacopeia. And this campaign and this kind of training program also we are going to expedite in a very systematic manner. The Government of India is working towards the elimination of anemia Mukt Bharat. The campaign, the rigorous activities are going on, and we have introduced the standards for ferric carboxy maltose, which is one of the injection based monograph, and this is the first time we have introduced in Indian Pharmacopeia, and it is going to help in the anemia elimination program as well. We are also equally committed to ensure the safety of vulnerable population. As per the data available, there are 20 universal immunization vaccines are used in the immunization program, but if you look at the current edition of IP, all the 20 standards have been developed. TB elimination, the Government of India is working towards the elimination of tuberculosis, and the standards, if you look at the standard development process, the Indian Pharmacopeia standards are at par with any other pharmacopeia when it comes to the TB. The bedaquiline, which is one of the multi-drug resistant tuberculosis medicine, which is first time introduced in Indian Pharmacopeia, and again the bedaquiline standards are going to be used by the TB elimination program. The bedaquiline, when it comes to the bedaquiline, the safety data through pharmacovigilance, Indian Pharmacopeia Commission is the first pharmacopeia commission globally, or first pharmacovigilance center to generate the safety data for bedaquiline. Similarly, when it comes to the standard also, we have introduced first time in Indian Pharmacopeia. Like trastuzumab, one of the monoclonal anti-human epidermal growth factor, again this is the first time we have introduced monograph in Indian Pharmacopeia. As I mentioned, the Indian Pharmacopeia beyond the borders. There are 20 countries have already recognized Indian Pharmacopeia globally, and many more countries are going to recognize through the diplomatic channel. So there is a huge responsibility for the stakeholders and the youth like you. You have a big responsibility to contribute and support in new monograph development as well, and also the revision and upgradation of existing monographs also. The Indian Pharmacopeia reference standards are the substances used for the characterization of our Indian Pharmacopeia monograph, and we are globally third in position. If you look at the Indian Pharmacopeia IP reference standards alone, we have 703, and we have 635 impurity standards are also developed. And it is, as per the regulation, it is mandatory for the manufacturer or the drug testing laboratories or any other stakeholders to use Indian Pharmacopeia and impurity standards in order to ensure the purity and strength of pharmaceuticals. in order to comply with the Government of India's mission to fulfill the digital, all the activities of Indian Pharmacopeia Commission, I mean the products and services of Indian Pharmacopeia Commission have been digitalized. We have online IP and then online digital tools for adverse events reporting like QR code, mobile app, online reporting, ADRMS. So all the products and services have been upgraded, digitalized, and I urge the stakeholders, students to use this facility particularly for adverse event monitoring and reporting, especially the QR code, which is being very well recommended and accepted globally for monitoring and reporting of adverse events. So I think if you look at the Vikshit Bharat 2047, the Indian Pharmacopeia Commission is moving towards significantly. There are some of the immediate priorities we have been addressing continuously. Most of the activities have been already addressed. The Indian Pharmacopeia 2026 is available, and the National Formulary of India, which is going to be released very soon. And then biovigilance to monitor the safety of organ and tissue donors and recipients will be launched very soon to monitor the safety of all those patients. So these are the immediate priorities. When we look at the futuristic aspects, when India is going to achieve Vikshit Bharat 2047, definitely the contribution from Indian Pharmacopeia also going to be significant, like the international footprint, Indian Pharmacopeia footprint to more than 50 countries, Green Pharmacopeia moving towards complete replacement of animals, inclusion of artificial intelligence in many of the monograph development process, and also the reaching of pharmacovigilance and material vigilance to the district level and even the primary health center level. These are the future priorities, and I take this opportunity to all the stakeholders to take part in the Indian Pharmacopeia journey with respect to promoting the quality and safety of medicines. With this, once again, thank you very much for this opportunity, and I wish you all the best. I don't know whether I can take any questions or not. Over to the organizers for further scientific proceedings. Thank you.
Thank you, sir, for such an insightful and enriching keynote address. We will now break for tea for 15 minutes. Kindly reassemble in the hall after 15 minutes so that we may continue the sessions on schedule. Thank you.
Owned by those who are powered by knowledge and wisdom that transcends time. There is one place where such wisdom is imparted, building individuals to build the world. Welcome to
Vinayaka Mission's Research Foundation, deemed university.
The university, with 27 constituent colleges, delivers cutting-edge knowledge across various disciplines. Medicine, engineering, architecture, management, health sciences, dentistry, measuring up to 135 courses of study, all covered across sprawling and student-friendly campuses. At Vinayaka Missions University, students of various disciplines hone their knowledge right up to doctoral levels. At any given point, there are 15,000 students studying in the campuses. With the vision of creating medical professionals who can meet the needs of the Indian and international communities, Vinayaka Missions University has made a significant contribution to the field of medicine. Four colleges and hospitals in Salem, Karaikal and Puducherry not only groom students into medical professionals, but also ensure easy and affordable medical help to the local population. Beyond medicine, the university is a multicultural hub that has students gaining expertise in a wide range of fields. Our engineering courses are designed to give students comprehensive knowledge in all contemporary fields of study. This is strengthened by state-of-the-art infrastructure. Engineering and technology apart, Vinayaka Missions University is known for its courses in dentistry, pharmacy, homeopathy, nursing, allied health sciences, physiotherapy, architecture, arts and science, and management. Our libraries are well appointed and are a digital and physical resource for both students and faculty. Linked to the central online catalogue, students have access to over 1,45,000 books, 1,000 journals, and over 5,000 audiovisual aids. owned by those who are powered by knowledge and wisdom that transcends time. There is one place where such wisdom is imparted, building individuals to build the world. Welcome to
Vinayaka Missions Research Foundation, deemed university.
the university with 27 constituent colleges delivers cutting-edge knowledge across various disciplines. Medicine, engineering, architecture, management, health sciences, dentistry, measuring up to 135 courses of study, all covered across sprawling and student-friendly campuses. At Vinayaka Missions University, students of various disciplines hone their knowledge right up to doctoral levels. At any given point, there are 15,000 students studying in the campuses. With the vision of creating medical professionals who can meet the needs of the Indian and international communities, Vinayaka Missions University has made a significant contribution to the field of medicine. Four colleges and hospitals in Salem, Karaikal and Puducherry not only groom students into medical professionals, but also ensure easy and affordable medical help to the local population. Beyond medicine, the university is a multicultural hub that has students gaining expertise in a wide range of fields. Our engineering courses are designed to give students comprehensive knowledge in all contemporary fields of study. This is strengthened by state-of-the-art infrastructure. Engineering and technology apart, Vinayaka Missions University is known for its courses in dentistry, pharmacy, homeopathy, nursing, allied health sciences, physiotherapy, architecture, arts and science, and management. Our libraries are well appointed and are a digital and physical resource for both students and faculty. Linked to the central online catalogue, students have access to over 145,000 books, 1,000 journals, and over 5,000 audiovisual aids. The hostel facilities at the campuses spell absolute comfort and provide the right ambience for self-study for the students. Our students are thoroughly encouraged in sports and cultural activities. Besides successfully participating in inter-university sports contests, our students have also excelled in cultural meets and festivals.
Vision for tomorrow, the mission of today, VMU. Bringing minds together in search of endless wisdom, VMU. Building individuals to build a world, building hopes and dreams on heart at VMU. Friends to stay, VMU.
Vinayaka Missions Research Foundation, Salem.
Welcome back all. We now resume the scientific deliberations with a session on navigating global regulations in clinical trials. It's a privilege to have us Dr. Melvin George, Center for Clinical Pharmacology and Center for Clinical Trials and Research, SRM Medical College Hospital and Research Center. Welcome, sir. I also welcome our chairperson, Professor Dr. J. Baby John, Dean, Vinayaka Mission, Sankaracharya Dental College, Salem. May I now request the chairperson to kindly introduce the speaker and conduct this session.
Ladies and gentlemen, it's my pleasure to introduce our next speaker of the day, Dr. Melvin George. To say a few words about his accomplishment, let me have the privilege of saying what he has accomplished through his career. Dr. Melvin George is a distinguished academician, clinician and a clinical research expert. He is currently a professor and head of the Center of Clinical Pharmacology and the Center for Clinical Trials and Research at SRM Medical College and Hospital and Research Center, and serves as a nodal officer for ICMR Indian Center for Clinical Trials. He is an alumnus of JIPMA, Pondicherry, where he completed his MBBS, MD in pharmacology and DM in clinical pharmacology. He has contributed to several national and international clinical trials, including Phase 1 to Phase 3 vaccine studies for COVID-19, dengue and influenza and has received multiple prestigious ICMR research grants. His academic records includes over 130 publications, 10 book chapters, more than 1,650 citations, and an H index of 24. In recognition of academic and research contribution, he was conferred the Most Promising Senior MD Pharmacologist Award in the year 2025. We really have a great person amidst us, and we are really honored to have you, sir. Please, your stage is for yours.
At the outset, I wish to thank the chairperson for the kind words of introduction, and special thanks to Dr. Jaker for the invitation to share my few experience in clinical trials, and also to the organizing committee of this wonderful conference. It's really a special moment because clinical trials very often is something that is misunderstood, misjudged, and if you look at the average newspaper items that come out of clinical trials, more likely you are likely to see a negative information about clinical trials than something positive about clinical trials in the lay press. And here at Vinaga Missions, we are having a full day session on clinical trials. So big congratulations to the organizing committee to have the thought process to put all these things together. I congratulate the scientific committee and the organizing committee to conduct a conference on clinical trials. How many of you watch movies? Raise your hands. Movies? Okay, only some of you. Others don't watch movies. Okay. So, in movies, there is a very famous person. I don't know how much youngsters are look up to him, but how many of you have heard of the word person by name Kamal Haasan? Kamal Haasan? Yes, all of you heard, right? Yeah. So, what is very special about Kamal Haasan when it comes to film making is, they say he is the encyclopedia of the film industry in India. Okay, a lot of sobriquets, a lot of special titles he has, and one of the unique things that Kamal Haasan is credited to doing is every latest technology from Hollywood. No, he is invariably one person who is credited to bringing that to Indian movie industry. Okay, some of you might have heard this. Why am I telling this story about Kamal Haasan right now is that just like how we are talking about the film industry looking at the US as the best place to get the latest technology and bring it to India so that Indian movies have as good technology as what we see in the West when it comes to movie making. Even in the drug development, if you talk about drug development as a whole, the gold standard for drug development is always the Europe and the US, particularly US. It's not that we do not know to develop drugs and devices. It is just that they've had a much longer experience, and most of the pharmaceutical companies are situated in US and Western Europe. As a result of that, a lot of development has largely come from the West. And when we talk about global clinical trial regulations, invariably the first and foremost group of regulations we will talk about is the US FDA regulations and the European Medical Agency regulations, because they are the ones which have set the standards for the rest of the world to emulate. So much so that recently, about a year or two, in India there was a guideline which came which said that if a new drug has been developed in certain countries, for example, US, Europe, and many other countries were mentioned, then the sponsor of that particular drug can request for waiver of clinical trials in India, because it is expected that if the drug has been developed in the US, it would have crossed a benchmark and there is no need to again sit and re-evaluate the drug in India, particularly if the drug is for a very important life-threatening disease where we want the drug to come to the market faster. So the point I'm trying to tell you is that many of our regulations that we have in our own Indian context, for example, the new drugs and clinical trials rules, Indian GCP, all these regulations, if you look at the foundation of these regulations, it is largely from the international regulations, okay, and the US FDA and EMA regulations have actually set a foundation for a context for the other regulations to emulate. So with that brief opener, I will now talk about EMA regulations and FDA regulations. The slides may be a little heavy, but I will just try to select certain points to make it simple, because I understand most of the audience is undergraduate, and I will try to make it simple enough for all of you to follow. So EMA, what is EMA? EMA stands for European Medical Agency. So if you all must be knowing about Europe. So Europe is basically, you have many countries within Europe, and currently the drug development in Europe is also centralized. What do we mean by that? It means that it is possible for a pharmaceutical company to get their drug approved across the different countries using a centralized process, because we have something called as the EU. Okay, so through the EU structure, the EMA provides the approval for new drugs in Europe. So a sponsor need not get individual country. Europe, when you talk about European Union, there are 27 countries in European Union. So a sponsor does not have to get approval from each of those 27 countries. He can get the approval from the European Medical Agency, and that is valid across different European Union states. So it operates under European Commission authority, and it is responsible for the scientific evaluation, supervision and safety of medicines. It coordinates network of over 50 national competent authorities across EU and European economic regions. And more than 100 medicines are evaluated every year. And if you look at EMA, the main method, the main portal that is used to evaluate all these medicines is Clinical Trial Information Systems, CTIS. It is the common EU portal for all clinical trial applications since Jan of 2022. So if you talk about European Medical Agency and its what are the key facets of this EMA's guidelines? Okay, we are going to talk about clinical trial oversight, particularly in the context of compliance to clinical trial regulations, clinical trial information systems supervision. We'll talk about the scientific opinion that the regulatory agency gives sponsors. For example, if I am a pharmaceutical company, Pfizer, and I want to develop a drug for bronchial asthma, before I venture into drug development, I can request an appointment with the European Medical Agency to help me and to guide me as to what is the expectation of EMA or European Medical Agency when it comes to the clinical trial drug development process. Okay, so the sponsor is prepared and he knows what is expected of him even before he starts the drug development process. So that scientific opinion, there is a centralized procedure available now. Of course, GCP inspections are largely driven by European Medical Agency. What is GCP? What is GCP? Anyone in the audience behind students? Good clinical practice. So what is this good clinical practice? Good clinical practice is the basic standard that one should follow while doing clinical trials. It is an ethical standard and it is a scientific standard. So only if you are doing that minimum GCP standards is the data going to be accepted globally. Okay, so there are people in the EMA who come and visit the sites, the hospitals which are doing trials, to see whether this GCP is really being followed. Okay, so that is another mandate of the EMA. And of course, guidelines. If you go to the internet, if you type EMA guidelines for drug development, you will have almost 50 to 100 guidelines on different aspects of developing drugs and clinical trials. And of course, guidance on pharmacovigilance and specific guidance on drug development pediatric population. So this clinical trial regulation, CTR, in EU, it came about in 2014, and it took, you can see the timeline, it took almost 10 years for this to become fully implementable even in an advanced country like Europe. So it's a lesson to say that it takes time for regulations to take full effect. Okay, so there's clinical trial regulation, it's called CTR. That is the current guideline that is to be universally followed if you're doing clinical trials in Europe. So within a period of 45 days, a decision is taken about any CT application. And if EMA does not respond, it means that authorization can be taken by default. Okay. Now, one good thing about this clinical trial information, clinical trial regulation is that it encourages transparency. It encourages transparency. So that means all clinical trial data should be publicly available on the EU CT register. There is something called as the EU clinical trial register. So you are supposed to disclose the results within 12 months of the trial end, and you are supposed to give a lay summary to the public, because at the end of the day all these trials are going to affect the general public, right? So a summary of the trials in lay language should be mentioned, available in the website. And the protocol, previously it was very difficult to access these clinical trial protocols, but now it says protocol should be present in public domain, and the summary of results also should be there. And of course I spoke about inspections and oversight that the EU will be taking. I'll not go into the details of that. And the EMA is supposed to also mandate annual safety reporting. Similar to a process we have in India called PSUR, we have drug safety update reporting, annual safety reporting that every sponsor should provide to the EMA at the end of every year, irrespective of the status of their clinical trials. So the application for a clinical trial is now a centralized procedure. So once the sponsor applies to EMA through the CHMP, that is the committee which evaluates these medical products, the drug is approved and it can be sold across Europe. Okay, and this is valid across Europe among all states. However, when it comes to pricing, when it comes to reimbursement, okay, see in India, who pays the money for medicines? Suppose a patient has to get medicines. How does he get the medicines? He gets the medicines mostly by out of pocket. He pays by himself. But if you look at Europe, most of the time it is they have insurance methods, they have insurance schemes which ensure that people get the medication free of cost through those insurance schemes, and many of them are public schemes, and these decisions are taken at the individual country level. So what is available in France through the insurance scheme might not be available in Germany. Okay, so those kind of differences you will see, because that is an individual country decision regarding how they are going to reimburse the drugs. Okay, and this scheme is applicable for all innovative medicines, for orphan drugs, and even biotech molecules. Of course, the good clinical practice guidelines, you all know, ICH E6 guidelines. It is mandatory to be followed for all EMA related trials, and the latest version of this ICH E6 guideline is the R3 guideline. Okay, R3, ICH E6 R3. So most of the things what you see on the left side, okay, these are all standard GCP principles. For example, protection of trial participants' rights, safety and well-being. Okay, every trial participants' rights, safety and well-being are more important than anything else. Okay, data integrity and credibility of the clinical trial results. So it is the clinical trial should be done in such a way that at the end of the day, anyone who looks at the data can actually trust the data. So the systems should be put in place that the investigator, the research coordinator, the ethics committee, the sponsor, the CRO, everybody follows the standard operating procedures for carrying out the clinical trial activities, so that you have credible data. And that credible data is only going to ensure a marketing approval. Responsibilities should be clearly defined for the different parties. For example, sponsor of a clinical trial has certain responsibilities, investigator of a clinical trial has certain responsibilities, and the ethics committee also has its own responsibilities. They are clearly demarcated and they are obviously expected to follow. Informed consent needs to be a fair process. It should be an ongoing process. What does that mean? It means that there is a possibility that after the trial is halfway done, we come to know of new developments. The amendment to the informed consent should be done and re-consent should be taken. Okay, and it should be a freely given consent. Obviously, we cannot take consent, force people to participate in trials. And of course, a sponsor and the site is expected to manage essential documents. All trial personnel are expected to have GCP training. Okay, so these are all general core principles that are universally followed and are the same even when it comes to Europe. So, what is specific or new in this GCP guidelines in R3 is what you see in the second column. Risk-based quality management. Risk-based quality management. What does it mean? Quality by design embedded at protocol stage. What does this mean? I am planning a protocol. I expect that when I write the protocol, there are some 10 to 15 points where there are critical issues that if I don't take care of it, the whole protocol is collapsed. For example, the way I measure the endpoint. For example, if an endpoint is occurrence of a major adverse cardiovascular event, imagine that is the endpoint of my study. Now, if I don't have a clear-cut method of how I am going to evaluate that major adverse cardiovascular event, obviously each investigator, each site, each hospital will have its own way of measuring that, and the data quality will come down. So we don't want to compromise there, right? So ensure that a uniform process is followed, that irrespective of site, whether the site is in Germany, whether the site is in Netherlands, whether the site is in Norway, the same method of reporting of the major adverse cardiovascular event is followed. So this is taken care at the protocol stage, not after the study is over and you start doing monitoring. By the time it's too late. So take care of this quality issues, okay, at the time the protocol is made. So this is actually a lesson for us when we do studies. Always anticipate problems where the problems are most likely to happen and concentrate on building those areas so that your trial or your study has the least likelihood of failure. Okay, so that is the concept of this risk-based quality management. I'll not go more into that. And of course, there are other sponsor and investigator duties that, as I mentioned earlier, has to be taken care of. And the ICH E8 guidelines is a general guidelines for all clinical studies, which also is to be implemented when designing clinical trials. Similarly, the adaptive designs are now more encouraged, because adaptive design is something like you do the study, start doing the study, but as you start doing the study, you realize that there are certain results that make you relook at the way you have designed your study. For example, you're comparing 4 different doses of the drug. Okay? You have started the study. At the end of 4 months of treatment, you realize that the 30 milligram dose is working, the 60 milligram dose is working, whereas the 10 milligram dose is not working. So there is no point continuing this trial with patients on 10 milligram just for the sake of completion. Rather, I modify my protocol such that those patients who are getting 10 milligram will get randomized to get 30 milligram or 60 milligram. In this way, I am ethically doing a good thing by minimizing the chance of failure and giving a futile drug to a patient. And in the end, I am getting better data of more important dosages, which is the 30 and 60 milligram dosage. This is an example to tell how an adaptive clinical trial design is something that is frequently utilized in clinical trial designing. Now we have something called as seamless phase 2, phase 3 design. Earlier thing was, I should first finish phase 1. Once I finish phase 1, I go to the regulator, get the permission to do phase 2. Then I go again after completing phase 2, I go again get permission regulator, then start phase 3. So that is a classical method of doing trials. Now you have something called a seamless phase 2, phase 3. So what does this mean? It means when I write the protocol, I will design phase 2 and phase 3 at the same time. I will complete phase 2. At the end of phase 2, I will have a DSMB committee. What is DSMB? Data and Safety Monitoring Board Committee. So that will evaluate the trial to see whether the trial can proceed as per the original phase 3 protocol, or should I make modifications, and of course get the ethics committee also approved for the subsequent trial, and straight away do the phase 3 trial. So the regulator gives the approval for both the phases right at the beginning, and he is only informed about the subsequent progress of the study. In this way, you are saving a lot of time. In fact, if you remember, in 2020, when we had the vaccine development for COVID, many of those trials followed the seamless phase 1, phase 2, phase 2, phase 3 design, so that drugs, vaccines could be approved at a faster rate. Okay, and the EMA guidelines also talks about estimand framework. So basically, it's a statistical term where we need to have clarity as to what exactly we are estimating when it comes to the clinical trial endpoints. And I'm not going to talk about DCTs because we had a very fabulous session by Dr. Mujibuddin for what DCTs are and how DCTs are rapidly changing the how clinical trials are evolving and becoming more efficient because of using DCT designs. And apart from this, there are many, apart from E6, there are many other guidelines which is mentioned on the right. I'm not going to the details of each, but suffice to tell you that these guidelines are for specific scenarios which need to be utilized when you are doing any EMA trial. For example, in pediatric population, in geriatric population, how do you do a dose ranging study? How do you provide safety reporting? Whenever a clinical trial happens, there is a likelihood of serious adverse events happening. So how do you manage that? All those things are mentioned. How do you choose controls when you design a study? Can you use placebos? Should you use a positive control? In which situation you should use placebo? In which situation you should not use placebo? So all these are clearly mentioned in these guidelines and need to be implemented appropriately. And then we have a detailed investigational medicinal product dossier, which needs to have all the preliminary data. Preliminary data means CMC, chemistry, manufacturing control data, preclinical data, and the clinical trial data until that prior trial. This is so important when it comes to reporting. Okay, we have adverse event, serious adverse events, adverse drug reactions, and SUSAR, suspected, unexpected, serious adverse reaction. So, what is a serious adverse event? Any reaction that is causing death, hospitalization, permanent disability, okay, congenital anomaly, we all know this. This is SAE. So, all SAEs have to be reported, and when it comes to SUSARs, they are reactions which are unexpected reactions, okay, and they are serious, okay, and these are reactions that have to be notified to the authorities within 15 days, and if it is death, you have to notify within 7 days. So 7-day, 15-day rule is what is followed in Europe, and of course apart from that we have regular annual safety reports being sent, and there are specific formats for it. And this last point I was telling, DSMB. DSMB is a committee which is appointed by the sponsor to review the safety of the clinical trial. So they are like a advisory committee which will tell the sponsor, is your trial safe? Can it continue or should the trial stop? Okay, sometimes the trial may be very safe, and so there is no no need to stop it, so you just tell continue. Sometimes the DSMB may say this trial requires the modification of the protocol, or sometimes the protocol will be, the efficacy will be so low that the DSMB may say there is no point continuing the trial, the efficacy is too low, even if this trial continues it is a futile attempt. So don't waste time, stop the trial. Or in the rare situation, a DSMB may also say this drug is doing super duper, it is unethical to continue this trial and give the control drug in the other arm. Let us not deprive patients of this drug. Let us stop this trial and allow this drug to be approved in the market, so that the drug comes earlier to the market. Okay, that is also a possibility. DSMB can come up with such recommendations also. Okay, I'll skip this. Okay, lastly I'll talk about scientific advice for EMA. So basically this is the mechanism which sponsors are given to consult on drug development strategy. So there are different methods. I'll not go into the details of that. And then now we talk about FDA guidelines. FDA is established in 1906 under US Department of Health. Over 18,000 employees, budget of $6.5 billion. Okay, I just tried to Google how much do we spend in India for drug development. It's around $400 million is what I saw. And when it comes to FDA, the guidelines is 21 Code for Federal Regulations. Code of Federal Regulations. This is the universal standard for clinical trials. Okay, and any trial done in USA should follow these guidelines. Is this applicable to us? You may be asking, why are you talking about US FDA guidelines, EMA guidelines and all this? The thing is, sometimes in India also we do clinical trials for which the data is going to the US FDA or the data is going to EMA. In all such situations, these guidelines are applicable even to us. We are also expected to follow some of these guidelines, of course without compromising on our own guidelines. So we have to follow Indian guidelines, NDCT. We also have to follow the FDA and EMA guidelines wherever appropriate. So don't think this is not applicable to India. Very often we do trials where the data goes to FDA. If that is the case, we have to follow the Code of Federal Regulations. So under FDA, there are again three categories: Center for Drug Evaluation and Research, which mainly looks at drug trials, different drug approvals. Then we have Center for Biologics Evaluation. So US FDA has a separate division for evaluating biologic molecules, that is all the large molecules, right? Infliximab, alemtuzumab, all those monoclonal antibodies, all those things will come under CBER. And finally, for medical devices, they have a separate division called Center for Devices and Radiological Health. So whenever a sponsor is trying to, 5 minutes? So I'm closing in 5 minutes. So whenever a sponsor is trying to send a application for developing a new drug, we call it as investigational new drug application. Okay, so a pharma company might submit a IND application for developing a new drug. Okay, and for that he has to submit a series of documents, and the main document is called Form FDA 1571. It is basically it's like a undertaking where he says, I'm going to follow all the guidelines of US FDA and I'm seeking drug approval. And once within 30 days, if he gets a response as his drug is an IND, he will get that IND status. But if he does not get a response, he does not get an objection, automatically his molecule becomes an IND. So that is the state of efficiency of US FDA. Sometimes the US FDA can say, stop, hold on, we are not convinced, there are certain things you need to do, that is called clinical hold. So they stop and they ask more clarifications. So in the interest of time, I'm skipping more details. The FDA regulations also talk a lot about informed consent process, the requirements for an ethics committee. Okay, the ethics committee evaluation is very, very stringent in USA. The minutes of the meeting is something that is very important. They have to really document that the product was really evaluated by the ethics committee before giving the approval to the clinical trial site. And we have different kinds of IND regulations in specific context. So just like how EMA is having specific advances, we have these kind of advances for FDA also now, like adaptive designs I told you. We have something called as master protocols. So master protocol means in one protocol, over a series of drugs can be evaluated through that single master protocol. And using Bayesian statistics, you can make your trials more efficient. And FDA is one regulatory authority which uses and has actually approved drugs purely on the basis of real world evidence. Okay, there are a number of drugs which have got marketing approval just on the basis of real world evidence. Real world evidence means data collected outside clinical trial context. On that basis also, real world evidence, the FDA has granted new approvals. And of course, the guidelines of the ICH, many of these guidelines are followed, are adapted even by US FDA. I'm not going into the details. Safety guidelines are also there. The same 715 rule applies. And I just want to focus on one thing, that there are specific designations called breakthrough therapy, fast track status, accelerated approval, orphan drug designation. Based on this, you have a chance as a sponsor to get your drug approved in the market at a faster rate than the usual rate. Okay, and after a trial is completed, the US FDA will come for inspection. If they find that there are issues with the way the trial is done, they give something called as a Form 483. Form 483. Form 483 says there were objections found in your clinical trial site. Okay. And if it is even worse, if the issues are too bad, they give something called as warning letters. Okay, warning letters are usually posted in the FDA website that this particular site, they had all these deficiencies. Many times you will find in the paper that this particular company was visited by FDA and they were found to have some objections in the way the manufacturing was done. So in the same way, even for doing clinical trials, they can give objections. And the worst part is debarment, disqualification of investigators. If they find there are perennial problems and it's repeating, they can totally debar investigators from doing further clinical trials and the site for doing clinical trials. So, to summarize, I just want to say that FDA has this 30-day safety review. They integrate the ICH E6R3 as one of the major plans when it comes to drug development. The form FDA 1572, it binds investigator to all GCP obligations. 715-day safety rule applies for FDA. And for pediatric studies, many times we don't have proper studies in pediatric population, right? So FDA is encouraging that sponsors come up with pediatric studies and really evaluate the safety of molecules in pediatric population. Okay, and there are special status like breakthrough therapy, fast track status, priority review for fastening drug development. So in the interest of time, I'm closing here. I'll be glad to answer if there are any questions. Thank you.
Thank you, Dr. Melvin. It was really a wonderful session because it was an eye-opener for clinical trials as such, because we are now into the process for most of our institutions, including our dental fraternity. And any quick questions from the audience? One question we can take because of the paucity of the time. Anybody? If not, I just have a doubt. You were telling about that adaptive design. So now if we have to change the design, do we have to again go back to the ethical committee approval for that?
Thank you, sir, for that question. So when it comes to adaptive design, usually the rules of how the adaptation is going to be done should be specified prior, not after the trial starts. So there are very clear-cut rules when you will make the change. And as long as the changes are made within that original rules, there is no need to again go back to the ethics committee.
Thank you, sir. And one more doubt, if you don't mind. Informed consent you were telling.
Yes.
So should it be audio or audio and video? This is a big confusion we have. So if we can highlight on it, we'll be happy.
So in the Indian context, audio-visual consent is expected to be done for all regulatory clinical trials which is done in vulnerable population. So if you're doing a clinical trial in India, you are taking a patient and that patient is likely to be considered a vulnerable patient, we have to take audio-visual consent. But if the patient is having HIV or if the patient is having Hansen's disease, leprosy, then the visual part can be excluded. The audio part alone has to be taken. Yes, sir.
Video coverage also, right? Thank you, sir. Thank you.
Thank you, sir, for such an insightful and enriching lecture. May I request Professor Dr. J. Baby John to present the memento to our speaker. May I also request Professor Dr. Gnanasekharan, Director, Innovation, to present the memento to our chairperson. We now move to the next session. I take great pleasure in inviting Dr. M. Kalaivani, Principal Scientific Officer, Indian Pharmacopeia Commission, as our distinguished speaker for the next session. Welcome, ma'am. I also welcome our chairperson, Professor Dr. Arun Daniel, Department of Community Medicine, Arubadi Veedu Medical College, Puducherry. May I now request the chairperson to kindly introduce the speaker and conduct this session.
Very good afternoon. Welcome you all to this pre-lunch session. I am indeed very happy and honored to present the CV of Dr. M. Kalaivani, who is the Principal Scientific Officer and Officer in charge of Biologics Division at Indian Pharmacopeia Commission, Ministry of Health and Family Welfare, Government of India. She holds a PhD in Pharmaceutical Sciences from the Tamil Nadu MGR University and has been associated with the Commission since 2010. She has played key vital roles in establishing Biologics Division and its state-of-art laboratory facilities, contributing to the development of India Pharmacopeia monographs for vaccines, recombinant DNA therapeutics, blood products, and alternative methods to animal testing. She collaborates with leading national and international organizations, including the World Health Organization and United States Pharmacopeia. A certified NABL assessor and accomplished researcher, she has authored around 65 scientific publications and contributed to multiple editions of National Formulary of India. Her works continues to strengthen the quality standards in biologics and support public health in the country. So, without much wasting of time, I request Madam to carry on with her session. Thank you very much.
Thank you, sir, for the introduction. So, I should say good afternoon, and I hope you had a long day, but I try to complete it very short, and I'll make it very brief, my presentation. And I was given with my slides, please. Yeah. Before going into my presentation, I thank Vinayaga Mission Management and the organizer and the Jaikar sir and Arul sir for giving me this opportunity. Thank you, sir. And my presentation topic will be on clinical trials for biosimilar monoclonal antibodies. So I'll make it because today the audience are students of PG, PhD and UG also. So I'll try to make it simple. Yeah. Thank you. And I'll try to make it simple and maybe we can make it more interactive also. So today's presentation, first I'll be telling in detail about the biotherapeutics, biosimilars and monoclonal antibodies. Without understanding these terms, we cannot understand further things in the clinical trial. And then I will not go detail into regulatory guidelines because all our speakers for today's session are talking about the regulatory guidelines of different regulatory authorities. I will not say in detail about regulatory guidelines. And yes, of course, I will be telling about development of biosimilar monoclonal antibodies and the preclinical studies. The factors needs to be considered in preclinical studies for developing biosimilar monoclonal antibodies, and the clinical studies in case of this monoclonal antibodies, and the requirement of post-marketing data. And of course, maybe it is out of topic, I want to introduce you young researchers into the term organoids, which so it is just an introduction, nothing more in detail. If it can influence even one or two researcher, it will be very good for the society. And then about the organoids in PKPD studies. So the first slide, as I said, I'll be telling in detail about biotherapeutics. So this slide shows the difference between the small molecule and the biological molecule. See, as we all know, the small molecules are synthesized in the laboratory, in the conical flask, whereas biomolecules are grown in the living system. So that is the difference between the biomolecules and the small molecules. So when something is grown in the biological system, that means if something is grown in our body, it has huge variations. So those variations needs to be considered when we work with these larger molecules. And in case of monoclonal antibody, most of us might have studied about hybridoma technology and monoclonal antibody production and all. So I am not going into detail about that. So these biotherapeutics are, as I said, these are very large molecule and these are all target specific and lesser side effect. That means it can be given for a particular target. It can most of the biotherapeutics are used for cancer treatments. Monoclonal antibodies are used for cancer treatments. So these are all target specific. When a drug acts in the target, it automatically reduces the side effect. And this mimics the body proteins because these are all protein in nature, and it, of course, somewhere or other it has few characters of bodily proteins also. And as I said, it is very large molecule, which is approximately more than 150,000 Daltons, the size of this molecule is, and it is highly complex and it never consists of a single moiety. And for example, here I have given for this biotherapeutics, like in case of therapeutic proteins, we can have as an example, most of us know about insulin. Insulin is everywhere today, so the insulin is one of the simplest example for biotherapeutics. And in case of therapeutic protein, we have erythropoietin, which is used in the treatment of anemia. And the important today what we discuss will be about therapeutic monoclonal antibodies. Here I have given an example of rituximab and trastuzumab. The name itself might be difficult to pronounce, but it is easier when we understand the nomenclature of these maps. And it revolutionized the treatment of chronic diseases like cancer, rheumatoid arthritis, psoriasis. So these, when we understand this molecule, when we go for the treatment with this molecule, it will give greatest efficacy. And there are peptides in the market, semaglutide and liraglutide, which are all like GLP-1 inhibitors. And nowadays, maybe in the newspapers and in the scientific literatures, you might have seen about semaglutide, which was approved for anti-diabetic, as an anti-diabetic drug. Now it is getting approved as an anti-obesity drug. So such things are there. So, but these are all very large molecule. So this is the difference in the size of these biotherapeutics. When we look at the aspirin, it is just, its molecular weight is around 180. Whereas when you look at the monoclonal antibodies, it has 150 kilodalton in size. So that difference will be there, that size difference will be there, and are all protein in nature. See, when we say about protein in nature, as we all might have studied during our immunology class, when a foreign protein enters into your body, it creates antigenic reactions, immunogenic reactions, right? So these are all protein in nature. When it enters into your body, it too has a immunogenic reactions, but those things are considered during the drug discovery, drug development phase, and those things are needs to be monitored during the clinical trial and all those things. And the difference shows the common example of insulin is very much smaller when compared to monoclonal antibodies. So this is the basic structure of monoclonal antibody. Monoclonal antibody is nothing but it is your immunoglobulin G, but it is monoclonal, target specific, antigen specific. So it has its the structure with the light chain, heavy chain, and its variable regions, and the antigen binding site will be there. Okay. It has its antigen binding site where that site is responsible for binding with the antigen, and it has a biological activity mediation. Once it binds with the target, then the activity starts. So the complementary region, FC region, is responsible for the activity. And the next slide, the next figure shows the 3D structure of these molecules. And yeah. And once again, here, as I said, which is in a simpler way, here it is given with an example of, I think, rituximab. But when you, as I said, the name itself, the rituximab, trastuzumab, the name, it has its own nomenclature which is given by international non-proprietary name by WHO. And when we say trastuzumab, it is 2, that means TU2. 2 means it is for humanized, and ZU, it is humanized, and XI somewhere, rituximab, XI, that means it is a chimeric one. So such type of differences you can understand, and these literatures are very well available in your, you can search anywhere. And as I already said, like this has revolutionized the treatment of life-threatening diseases and chronic diseases like cancer and solid tumors, rheumatoid arthritis, psoriasis, etc. And it has the huge potential. By 2023, it is expected to flood in the market with the, and it has the market potential of $800 billion by 2023. So that means your small molecules will be replaced with the biological drugs in future. And these are all list of monoclonal antibodies which are approved in India. And yes, we understood what is monoclonal antibodies. Now we should understand what is biosimilars. Why we are telling biosimilars? Why not the generic alternative? What is the difference between biosimilar and generic? So that we need to understand. For example, in case of paracetamol, if I make paracetamol, if you make paracetamol, it will be the same paracetamol only. Will have a same structure. Whereas in case of biosimilar, there might be minor differences in the biosimilar. If I make one biosimilar, my biosimilar and your biosimilar will be different. There are minor differences, but those differences should be clinically inactive in terms of safety and efficacy. So the definition says biosimilars are legally approved subsequent version of innovator biotherapeutic product made by different sponsor or manufacturer following patent or exclusivity expiry of innovator product. So these biosimilars are produced, or these biosimilars are developed once the patent of innovator product is expired. And these biosimilars are having structural and manufacturing complexities, because of which there are minor differences are there. So they are not considered as a same, they are considered as a equivalent to innovator biotherapeutic. So here I have given an example of rituximab itself, where Mabthera, which was an innovator rituximab by Roche, after the patent expiry, there are biosimilars which are available in India, Reditex and Ritucat. Both are rituximab only, but they are biosimilar version of innovator product. The first who makes that product is considered as an innovator product, and following that innovator product, if someone makes a product of that, then that is considered as a biosimilar. And since it is considered as a biosimilar, the clinical trial requirements varies when compared to the generic molecule. So the second example which I have given is trastuzumab, where Herceptin is the innovator molecule invented by Roche, and then it is followed by Canmab, Hertes, etc., which are all biosimilar brands which are available in the country. And there are various terminologies used globally, and generally it is said as biosimilar, whereas in India it is said as similar biologics and similar biotherapeutic products in the WHO guidelines. As we can see, these terms follow on biologics in certain US FDA guidelines initially, but nowadays those are all considered as a biosimilars, and then subsequent entry biologics in Canada. So all these terms are synonymously used globally. And this is the basic definition of biosimilar as per the regulatory documents it is shown here. And whereas in India it is said as similar biologics, which means, see, I request the students to please listen. If it can influence you, even a single word, it will be helpful for your future. I may not expect you will be understanding everything what today it is said, but a single word can influence your future. Yes, so like in India, similar biologics means biological product which is similar in terms of quality, safety and efficacy. So it is similar in terms of quality, safety and efficacy. In comparison to reference biologic product. So reference biologic product means, does it need to be approved in India? No. It may be approved in India or it may be approved in ICH countries. So that provision is given in the India definition. Whereas in case of USA, same, highly similar to reference product, notwithstanding minor differences in clinically inactive components. But these reference products needs to be approved in US FDA, and that has to be compared if someone want to develop a biosimilar product for US. And whereas in case of EU, the similar type of, if someone want to develop a biosimilar for EU and that innovator product or the reference product needs to be approved, needs to get its approval from in EU, and that needs to be compared for biosimilar development in European Union. And as it is already said, there are various advantages of biosimilars. It reduces the overall healthcare cost and it is more affordable. If I say if it is more affordable, it is not like something comes for 100 rupees or 50 rupees. It also costs in the lakhs for the complete therapeutic cycle. But when compared to the for the innovator molecule, the cost of the biosimilar drastically reduced to 50%. So such healthcare affordability is there in case of biosimilars. And it has and it, of course, when there are a lot of manufacturers, it improves the global access among the patients, and it meets the growing demand for biologics. Growing demand nowadays, when we roam wherever we go, in every city we can see the cancer hospitals. So there is a huge demand for these biosimilars and biotherapeutics. And then it is already said it is used for chronic diseases. And where the difference is in terms of, I'm telling, there is a difference between innovator product and biosimilar product. Why there is a difference and where that difference is? When you look at the structure on the structure over there, the small molecule is having the same structure. It is shown there, which is a generic example of acetaminophen is shown there. So the innovator molecule and the generic version, both will have same structure. There will not be even minor differences also. Whereas in biomolecule, there is a smaller difference, minor difference in the maybe that amino acid sequence. So that shows that difference will be there between the biosimilars. That means if I am making an innovator product, if you are making biosimilar 1 and another making is biosimilar 2, all the three will not be same. All the three are similar 1. So that similarity will be there but not same. Why the differences are there? Because though products are same, vector which is used for production of these monoclonal antibody or these biotherapeutics are different, will not be same as what innovator is used. And the host cell expression, different host cells are used by innovator and as well as by the different biosimilars. So there comes the difference, because the host cell is the biological, it is a biological system, it is a living system. And then the production of protein will be same, both will make rituximab in case of rituximab, but and the protein recovery technique will be different, purification technique will be different, and the characterization of protein, different methods are used for characterization of protein. So the difference between innovator product and the biosimilar product is because of the vector which is selected and because of the host cell expression, host cell which is used for expressing that particular protein. And these are all various guidelines. The links are given. Every regulatory authority is having their own guideline. There are n number of guidelines. So that needs to be read thoroughly before going into anything. And this is just a regulatory framework for the approval of all these things. The world regulation, like you need to refer the WHO documents, TRSs, ICH guidelines, etc. Whereas in India, for these biotherapeutics, we have regulation of RCGM, CDSCO, State Licensing Authority, and Central Drug Testing Laboratories, Central Drug Laboratories. And other regulations in India covers, like we have NPPA for pricing of any molecule, and Environmental Protection Act, because these are all biological in origin, and then DBT, DST approvals are required, and of course since we work with animals, CPCA guidelines are needs to be referred. And in case of quality standards, we have pharmacopoeias. Every country will have their own pharmacopoeia. And this is the general flow chart for monoclonal antibody development, where the monoclonal antibody, like we need to understand the biosimilarity principle, establish the biosimilarity, and the reference product selection is important, which needs to be selected as per the regulatory guideline, and the cloning and cell line characterization is important. Process and analytical deviation, where it differs, that needs to be mentioned. And in case of biosimilar development, comparability exercise needs to be established. So that comparability, I'll be telling in detail where that comparability exercise needs to be established, and the process characterization and process validation needs to be taken care. This I have taken from Indian Pharmacopeia only. So if you have Indian Pharmacopeia IP 2022, you can see these flow charts. And this is what the biosimilar development, the important slide to understand the biosimilar development is this. Here, as I said, we need to compare the biosimilar with reference biological product. So in terms of what it needs to be compared, in terms of analytical characterization, extensive analytical and functional characterization are required. And this gives the structure and physiochemical comparability. When we talk about protein, it has primary, secondary structures and all. Those comparability needs to be established with the reference biological product, and higher order structure also in case of proteins, and functional and biological characterization by using in vitro in vivo assay. So it needs an extensive comparative, analytical and functional characterization is required. And in case of non-clinical studies also, it's need to be done comparatively. Comparatively means the data which you develop with the biosimilar, that needs to be compared with the reference biological product. So everywhere this comparative analysis is required. And in case of clinical PK/PD studies also, comparative clinical studies needs to be done. And in terms of safety and efficacy also, the comparative exercise needs to be done. The comparison is done only against the reference biological product. What is reference biological product? It is the innovator molecule. And there is a term called interchangeability. See, when someone establishes, if there are 4 manufacturers for paracetamol, we can interchange. Today I can take the paracetamol of one manufacturer, tomorrow I can take another. Nothing will happen to the patient. But in case of biosimilar, it should not be interchanged. If a patient is taking an innovator biotherapeutic product, that needs to be continued with innovator molecule only. If someone is taking with biosimilar 1, then that should not be interchanged with biosimilar 2 or 3 or with the innovator product also. So that interchangeability approval needs to be taken from the regulatory authority, and this approval happens only with US FDA, not anywhere. In India also, every product is considered as a new drug. Yes, once again I said, biosimilar have no clinically meaningful difference in terms of safety and efficacy in comparison to reference biological product. Reference biological product is nothing but an innovator product. The innovator product is nothing but the one who comes with that molecule first and who got the patent for that product. That is innovator. and as per the guideline. So after this, I will be talking about the Indian guideline, which is available, similar biologics guideline, which are available for the development of similar biologics. The first and foremost, the important step is selection of reference biological product is important, and as I said, it needs to be the reference biological product or the innovator molecule needs to get approval in India or ICH countries. So those molecule can be considered as a reference biological, and against that reference biological product, biosimilars can be established in India. And before preclinical studies, before initiation of preclinical studies, these are all prerequisite, and you need to go for RCGM approval. Before going for in the application of RCGM approval, it needs to, the application should have institutional biosafety committee, and the approval of institutional biosafety committee and the approval of institutional animal ethics committee. These approvals needs to be accompanied with RCGM application, and the application of RCGM, it should have the data on consistency of the product, process, product characterization and product specification. So these and basic clinical informations and preclinical study protocol, these needs to be submitted for RCGM approval. After RCGM approval only, toxicological studies needs to be initiated. And in case of preclinical studies, where efficacy and toxicity are done. As I said, I will not talk detail about the clinical trial because other speakers are there to talk about this. I will say the key words only. And here, these preclinical studies are conducted in the final formulation of the biosimilar, and it needs to be compared with reference biological product. And the dosage form, dose, strength, route of administration, all these information should be same as that of reference biological product. And this in vitro, in vivo studies can be done in the preclinical, but in the relevant animal species. And at least one repeat dose toxicity study in the relevant animal species needs to be conducted. And in case of clinical studies, we have PK/PD studies, pharmacokinetic and pharmacodynamic studies, confirmatory safety and efficacy studies, safety and immunogenicity data. Here immunogenicity data is required because these biosimilars are protein in nature, it may create immunogenic reactions. And extrapolation of safety and efficacy data to other indications. This is about the PK/PD studies, pharmacokinetic studies. It has, you need to refer CDSCO's guideline on bioavailability and bioequivalence also. Not only similar biologics guideline, this guideline needs to be referred, and this PK studies all needs to be, every study needs to be a comparative one with reference biological product. And the pharmacodynamic studies which are done in the patients or in the healthy volunteers, whereas in case of high risk molecule it is only in the patient. And this is about the PD studies where the PK/PD studies are most sensitive to detect the differences between the two molecules, and it gives the dose justification, and it may reduce the need for larger efficacy trials. That provision is also there as per various guidelines are there. And PD markers plays a key role here in the pharmacodynamic studies, I think, which is similar to that of other molecules also. But in case of, see for example, here I have given an example. What will be the, see we need to go for a right clinical endpoint here. Where in case of adalimumab and infliximab, reduction in CRP level is the endpoint. So such type of endpoints needs to be selected for this pharmacodynamic studies. Here I have given those as an example. It gives the similarity in the mechanism of action between the reference biological product and the biosimilars. And the confirmatory safety and efficacy study, say after preclinical and PK/PD studies, these studies are done, and the safety and efficacy study, once again, these needs to be compared with the reference biological product. but it needs to be done in the sensitive patient population, and clinically relevant endpoints needs to be selected. For example, here as an example of trastuzumab is given. As we all know, trastuzumab is given for the treatment of breast cancer. So here the sensitive patient population will be women with HER2 overexpression. So that will be the sensitive population. The study needs to be done there. And whereas clinically relevant endpoint in case of trastuzumab for efficacy, the clinically relevant endpoint will be absence of residual invasive tumor in both the breast and lymph nodes. Whereas in case of safety, endpoint for safety is reduction in left ventricular ejection fraction greater than or equal to 10% from the baseline or less than 50%, or symptomatic cardiac dysfunction. So these are all the clinically relevant endpoint to establish the safety. And of course, during these studies, nature, severity and frequency of adverse events also needs to be compared with the reference biological product. And safety and immunogenicity data needs to be established. Safety data, as I said, and immunogenicity data, where the point needs to be considered as anti-drug antibody incidences. As these are all protein in nature, when your body develops antibody against these protein, then efficacy will not be there, because your body will work against that. So there will not be efficacy at all. Anti-drug antibodies will be developed. So such type of incidences needs to be monitored here. And similarly, neutralizing antibodies needs to be monitored, and it needs to be compared with reference biological product. And extrapolation of safety and efficacy data for other indication. There is a question, if a study is done for one indication, can we extrapolate that for other indications also? Yes. At what conditions? When similarity with respect to preclinical assessment is established, clinical safety and efficacy is proven for one indication, and mechanism of action is same for other indication also, then the indication can be extrapolated. And for example, here once again trastuzumab is given in case of innovator or reference biological product, and in the biosimilar it is stated here, investigated only in metastatic breast cancer, but it is approved for early breast cancer, metastatic breast cancer, metastatic gastric cancer also. So when mechanism of action is similar, when the preclinical assessment and clinical safety and efficacy are proven or compared with the reference biological product, it can be extrapolated for others. If a biosimilar is having newer indication, it goes for a separate set of guidelines and all. And there are provision for waiver of safety and efficacy on all these conditions when structural and functional comparability are compared at the higher level up to the satisfaction of regulatory authority, and the preclinical evaluation is done up to the satisfaction of regulatory authority, and comprehensive post-marketing risk management plan is there, then the safety waiver can be given case-to-case basis in case of safety and efficacy trials. And for post-marketing data, you need to have a risk management plan which should cover a pharmacovigilance plan, adverse drug reaction reporting, and post-marketing studies which are similar to that of the small molecules, which maybe, yes, like in the pharmacovigilance plan, periodic safety update reports needs to be submitted to the regulatory authority every 6 months, and for first 2 years, and annually for another 2 years. And adverse drug reaction, if any serious unexpected adverse reactions are monitored, that needs to be reported immediately within 15 days. And the post-marketing studies should be, there should be a complete plan for post-marketing studies also. Yeah. So that's all about the clinical, the development of biosimilar monoclonal antibodies. As I said, I'll introduce you to the organoids. So here it is, organoids are nothing but a 3D structure derived from the stem cell. They can replicate the spatial organization and tissue specific function of real human organ. So when real human organ can be established at the laboratory level, it can replace the early clinical trials. That means the use of human in the early clinical trial can be avoided. So that advantage is there. So the researchers, those who want to go into the basic research on biological system can think of organoids and all. And why organoid is important? Because in case of biologicals, 90% failure rate is there. That means 90% of the molecule which clears till preclinical level fails at clinical level, because at preclinical level safety and efficacy are established by using animal model. There are differences. Being a science students, we can understand the difference between the human and animal. So that differences makes it fail when it goes for clinical studies. So that 90% failure rate is there. So during those development, it incurs a cost, and when there is a failure, where that cost will go? Automatically it will come to the patients. So these organoids can reduce the failure rate of drug development. And there are regulatory approvals also given. US FDA had given this amendment in 2022. Organoids can be used during preclinical and early clinical stage, whereas in India also we are coming up with the guidelines. Very soon, I think draft guideline is there. So that consideration is there in India also. Yeah. Yes, in my concluding remarks, as I said, monoclonal antibodies are target specific and with lesser side effect. And this is the cost effective option when compared to innovator biologics, and it increases the global access for this molecule to the patients. And how it is established? Based on totality of evidence approach. At every stage, comparative, analytical characterization, clinical, preclinical, all the studies needs to be established comparatively with reference biological product. And organoids can emerge as a human relevant tool which can reduce the uncertainty of the drug discovery, and it will be a step forward towards the ethical, efficient, patient-centric biosimilar development. And of course, harmonized regulatory acceptance is the key, and that will also come in the future. Yes, that's all about the topic for today. Since I came from Indian Pharmacopeia Commission, if I am not talking about Indian Pharmacopeia Commission, that will not be good on my part, and it is already explained by our director sir also. But Indian Pharmacopeia Commission, the name reflects, we not only publish as Indian Pharmacopeia and supplying the reference standard is the primary mandate. Apart from that, we publish as National Formulary of India to promote rational use of medicine, and we coordinate pharmacovigilance and materiovigilance to monitor safety of drugs in the country, and we do give skill development programs, and we do encourage the research activity at our commission. That means Indian Pharmacopeia Commission works for quality, safety and rational use of medicine. For more detail, kindly visit Indian Pharmacopeia Commission website, and if anyone interested for doing any of your research with respect to the analytical part or in the safety related part, you are always welcome, and the research can be done at the Indian Pharmacopeia Commission. And you can reach me at this email ID, and our website is given here. Yeah, thank you for listening me, and I know this is very late in the morning session, but I tried to reduce my presentation. Thank you for that, and once again I thank the Vinayaka Mission Management for giving me this opportunity. Thank you, sir. And yeah, any questions? I am ready.
Thank you, Dr. Kalaivani madam, and it was excellent and very crisp talk on biosimilar molecules for monoclonal antibodies. And I was quite impressed with the statement made that it will reduce 50% of the cost for monoclonal antibodies. And we could see two molecules there. One is the semaglutide, which is weekly once insulin, and it has given the liberty of not pricking every day for the patient. But when it comes to the cancer drugs like rituximab, being from the regulatory part, why can't the molecules which are available less of cost, like biosimilar products, why should they wait till the patent expiry has to go on? Why can't they come to the market earlier and save a lot of cost for the patient?
Sir, actually, your question is right, but for a person, for a company to develop an innovator molecule, here I have shown the data which are required for biosimilar development, whereas in case of innovator product development, there are huge data with respect to not only the analytical characterization in the clinical study, preclinical study, everywhere there are huge data needs to be established, study needs to be done, which incurs a cost. To till that cost is needs to be, like they need to take it back. So till the patent expiry, this is not, but in India, in case of trastuzumab, emergency approval was given by considering the importance of trastuzumab for the treatment of breast cancer. That steps are also be taken, but on the case by case basis.
Thank you so much, ma'am. And any questions from the audience? You can take one. If there are no further questions, I thank Madam for an excellent lecture, and thank you very much, ma'am.
Thank you, ma'am, for such a comprehensive and highly informative presentation. May I request Professor Dr. Arun Daniel to present the memento to our speaker. May I also request Dr. M. Kumar, Principal, Vinayaka Missions College of Pharmacy and Organizing Associate Secretary, to present the memento to our chairperson. Thank you, sir. We shall now break for lunch. Kindly reassemble by 2:15 p.m. sharp as we continue with the post-lunch scientific sessions.